My name is Emma Waters, and I would like to tell you about an adapting world from my point of view as an LGBTQ+ genome scientist. Now, firstly, I will give you a bit of my scientific background before I tell you about the last 30 years of genome sequencing and LGBTQ history in the UK. First, I'll give you a bit of my scientific background. So, I'm originally from Norwich, and throughout my career I've spent a lot of time at the University of East Anglia and the Norwich Research Park. My scientific journey began during my A-levels, where I got a summer placement at the John Innes Centre, which showed me what my dream job was. I then spent 4 years at the University of East Anglia doing a Masters in Chemistry before I started a PhD in Biochemistry. During my PhD, I investigated how to attach tiny chemical solar panels to bacteria so they could harness solar energy to produce useful chemicals like hydrogen or other fuels. After my PhD, I did a few postdocs and changed my research field to medical microbiology, and now I'm a senior researcher at the Quadrant Institute. This picture of me performing sequencing on a clinical bacterial sample sums up my day-to-day work. All of my work in Norwich has given me loads of opportunities to work in other places with people with a huge variety of backgrounds. In the UK, I've worked in Leeds, Cambridge and London. And then outside of the UK, I've worked and collaborated with people from Los Angeles and Montreal, all the way to Australia and Fiji. So now I'd like to tell you about genomic sequencing, which lets scientists read the life-giving instructions contained within the DNA of organisms. The history of genomic sequencing is all really recent, with a lot of it happening within my lifetime. It began in the 70s when scientists developed a technique called short-read sequencing, which was used to sequence the first full genome of a virus, which was around 5,000 letters long. Then the first big thing in my lifetime was the launch of the Human Genome Project, which began in 1990. Where a group of scientists wanted to determine the 3 billion letters of human genome, which was only achieved after 13 years of hard work. In that time, scientists sequenced the genomes of other organisms, each time celebrating because they sequenced something bigger. Going from 1985, where the first bacterial genome was sequenced, which contained a couple of million letters, and then 5 years later, when a fly genome was produced, which contained a couple of 100 million letters. In all this time, sequencing computer power kept improving, and in 2008, only 5 years after the Human Genome Project had ended, another Human Genome Project was launched with the aim to produce thousands of human genomes. In just 2 years, over 2,500 human genomes were produced to start to understand genetic diseases. In 2014, Public Health England started to use short-read sequencing to manage infectious diseases, which was essential to monitoring variants in the current pandemic. So how does sequencing allow scientists to figure out the instructions of life? Imagine this awesome rainbow-coloured diagram is a genome, with each colour representing a different gene, which provides unique instructions on how to build a different protein. Each gene here contains 1,000 letters of genetic code, which is perfect for short-read sequencing as it produces reads containing 100 to 1,000 letters long. Scientists can then start to put together a puzzle of what is in the genome by overlapping the sequencing reads to identify what coloured genes are next to each other. Short-read sequencing is highly accurate and can identify small single-letter changes in the genetic code in bacteria and viruses. The importance of sequencing was highlighted in last year with the COVID Grinch of 2020. COVID-19 contains 30,000 genetic letters in its genome, 4,000 of which encode the spike protein which the virus uses to attach and invade other cells. Scientists in the autumn of last year spotted that two of these letters had mutated and changed the instructions which encode the spike protein, allowing it to potentially become more contagious. And this was proved to be true when the government confirmed it was more contagious and locked us down for Christmas. So I hope you can appreciate how useful short-read sequencing is at spotting single DNA mutations. However, short-read sequencing is unable to resolve long repeat sequences, like here where we have this rainbow pattern repeated in one gene. And the short reads are confused on where to map to. I use a technique called long read sequencing, which produces reads that are thousands to millions of letters long. And this type of sequencing can identify a different sort of mutation where genomes can rearrange large sections of their genomes to shuffle the order of their genetic instructions. Now, this type of variation allows bacteria to go asymptomatic and potentially transmit silently and hide full detection from us. So now to switch stories altogether, I'd like to tell you about my LGBTQ+ journey. Now, I class myself as bisexual, but you could also call me a pansexual as well, as I believe as long as you are happy, you can be with whoever you want to be. In my A-levels, I had my first LGBTQ+ relationship with a transgender individual, but I only started to come out to my friends when I was in my late teens. Then when I was 25, I fully came out to my friends and family when I started to date a beautiful woman who I'm now proud to call my wife. I've never been ashamed of my sexuality, but why did it take me so long for me to fully embrace it? And it was only when I looked at the history of LGBTQ+ in the UK that I realised something. A big thing that overshadowed my childhood was a law called Section 28, where educational resources which promoted the alternative gay family lifestyle were censored, and teaching in any school of the acceptability of homosexuality was banned. This meant there was no LGBT role models for me to look up to when I was growing up. And in response to this Section 28, Stonewall was founded in 1989, and they fought really hard for our rights. After Section 28 was abolished in 2003, it took another 6 years for the government to apologise for the disgusting law. But since the law disappeared, our rights have improved greatly, with things like equal rights for civil partnerships, trans people being given legal recognition in their appropriate genders, same-sex couples being allowed to marry, and the unturning law which pardoned all historic instances of gay criminal convictions. Now, these important changes in our history has impacted generations of LGBTQ+ individuals in many different ways. I'm just going to give you 3 different examples which have occurred in my own career. In my early career, I was fortunate enough to be a part of an awesome group, but we all shared one common secret from each other, and that was that we were all LGBTQ+ in one way or another. It took the younger members of the group over 1 year to come out to each other, and this is probably because we were all from the generation which grew up during this Section 28 law. The older member of our group has still never properly come out to us, and this is probably because when they were growing up, it was illegal to be gay in the UK, and also probably because at the beginning of their career, it was hard enough for a female to be in science, let alone an LGBTQ+ female. In my mid-career, after I'd fully come out, I met an individual who was originally from another country, which had different religious views to ours. And after a month of working with us, this person still didn't know if it was okay to say that they were gay until I was talking about my future wife. And it broke my heart to see them in that moment because they looked so relieved that they could be themselves fully. And now I'm so happy to be part of the Quadram Institute and the Research Park, where every type of person and individual are welcomed and celebrated for their own uniqueness. And because of this open environment, everyone is comfortable to be their full selves. We have older individuals coming out like they never have before, because the next generation is so happy with who they are. And I know I'm speaking from my own experiences, but generally, scientists, even from countries where it is still illegal to be gay, doesn't care who you are or what your sexuality is, as long as you are happy and produce the best science you can. So why are Pride events still important? Even though a lot has been done in the last 50 years, which has improved the lives of many LGBT individuals, there's still a lot of work to be done. So if you look at the global image, there is actually still 69 countries where it's considered illegal to be a homosexual, and 9 of them actually incur the death penalty for those individuals. Today is LGBTQ+ in STEM Day, and the reason that this is such an important day is there's a similar image that is seen in science. So a lot of progress has been made again for our community in science, but a lot more can still be done. A recent survey which questioned LGBTQ individuals in STEM was carried out by the Royal Society of Chemistry. Within this survey, 28% of LGBT+ respondents said that they sometimes considered leaving work because of the climate or discrimination they had towards against them. And there was also 20% of trans correspondents which considered leaving often because of the discrimination they faced at work. This is why LGBTQ+ in STEM Day is so important, because science should be for everyone